Hello students. The topic of discussion today is post-transcriptional gene silencing. It is also called RNA interference. Okay. Basically, um, this results in downregulation of the gene at the RNA level. So the RNA, so transcription is over, RNA has been made, and then somehow the translation is blocked or stopped from happening. Okay, we will see why, and that's how it gets downregulated. Now it has been observed to be heritable. However, it is very dynamic and can be modified in subsequent cell divisions or generations. So it's a dynamic process. How it is achieved, we will see a little later. Now, prior to this siRNA mediated knockdown strategies, we have been heavily relying on antisense technology. So from 1980s to repress specific genes, because in higher organisms, in E. coli, in lower eukaryotes like yeast, you can create gene knockouts pretty easily. Okay, there are a lot of genetic manipulation techniques which are available. However, for higher plants and animals, it is very difficult to get gene knockouts. So these antisense technology provided an alternative means by which you could downregulate or lower the expression. That was called the knockdown approach. Knock, and the idea was so normally the protein coding RNA is called the sense RNA. So antisense RNA is just the production of the complementaries. The sequence, the RNA will be complementary to that of the sense RNA, as a result of which it will go and hybridize with the sense RNA, and that will prevent the translation from happening. So basically, it is to convert the sense RNA into a double-stranded RNA and block the translation. That was the main idea. After the discovery of this siRNA or RNA interference, now in hindsight, we feel that when we were trying to knock down using antisense RNA, that also probably led to this downregulation by inducing the RNA interference rather than actually what we had assumed that it will go, and because of blocking the ribosome docking site, it was inhibiting translation. So in most probability that also this antisense technology was actually working by inducing what we will be now talking about, that is the RNA interference. So RNA interference was first discovered in plant system in Petunia, when there was an attempt to kind of increase the pigmentation or anthocyanin pigment by overexpressing, by providing some transgenes that are responsible for the synthesis of these pigments. Okay, so it was discovered by this person, and in the 1990s, and what they had observed actually when they tried to re-engineer and put a transgene into the Petunia, okay, um, with the transgene had a lot of homology to the endogenous Petunia gene. So he observed something which was very peculiar and opposite to what he wanted to achieve. Okay, so the desirable effect was like a higher level of pigmentation because you are increasing the level of the pigment producing gene. But what happened was the pigmentation was kind of lost, and both the endogenous gene as well as the transgene looked, when you tried to measure the transcript levels, when you try to look at the expression level, you found that they are down regulated. There is hardly, it seems to be suppression. Okay, so it was also this phenomena at that point of time was referred to as a co-suppression, as a providing a gene which is homologous to the endogenous gene. It was expected to increase the level, but it resulted in the decrease in the amount of the actual uh, pigment producing genes. Okay, and what happened, what was observed when they looked into the molecular basis, it was observed that there was mRNA was degraded. Okay, both from the endogenous as well as from the transgene. So the sense mRNA was cleaved off, was degraded. Okay, So instead of turning on, the organism turned off not only the transgene but also the normal cellular copy of the gene. So this is basically, as I said, they involved attempts to manipulate the pigment synthesis genes. So they are involved, mostly the enzymes that are involved in the flavonoid and anthocyanin pathway. This was one of the gene that they tried, and another gene, this is the reductase. So this is the gene. We will see some results with the DFR transgene introduction into the system. And what was observed that when these genes were introduced in the petunia using a constitutive, a strong viral promoter, okay, both the mRNA levels as well as the phenotype, okay, that is the pigment levels, were dropped in many, like in 25% of the transgenic plants. We saw that rather than overexpression, we saw it was further reduction. So again, we don't need to get into all this, the pathway. There are different, this chalcone synthase is here, DFR is somewhere here, and they give rise to basically strong pigment, anthocyanin, strongly pigmented compounds. In petunia, this does not happen. The orange color pigmentation does not happen because this precursor is not recognized, okay, as a substrate by the DFR enzyme. But the other two, that is the red coloration, cyanidine derivatives, and the this particular, which is purple derivative, okay, delphinidine derivatives. So they are made, so you can get purple color petunia or you can get red color petunia petals. Okay, so they wanted to increase the intensity of these coloration, the red coloration and purple coloration. So what they did, so this is like after the transgenic. So this kind of plants were observed. Okay, so there were patches of white in the red. There was then complete lack of pigmentation. Then you had this purple with the patches of white where you hardly see any pigmentation. Okay, so as I said, in at least 25% of the transformed plants, they were like white or patchy purple and white, opposite of what they had intended. They intended a very, very strong pigmentation. Instead, they got this kind. Now, what was their construct? So basically, this was kind of the construct where they had the DFR cDNA and with a strong constitutive promoter, okay, called mosaic virus promoter, a very strong promoter, so had high expression from this cDNA construct, and also nopalin synthase, so 3' processing signal, because you are putting it in the plant, so that's why you had used some of the plant regulatory signals. And using this construct, when they introduced it, this is what they observed. Okay, so and from the non-pigmented area, they saw that the mRNA levels for DFR, although there was a transgene and the endogenous gene, still the mRNA levels were pretty low. However, the transcription was not a problem. So if you actually monitor the transcription initiation, then that was not a problem, but something was happening to the mRNA after the transcription. Okay, so this is almost resembling that what an antisense technology, like using an antisense DFR gene, if that would have done, then what would have been achieved? Same thing was achieved with a sense construct. Okay, so this was supposed to be giving you overexpression, rather it gave you down regulation. Okay. Now, this is not that exclusively only in plants, but later on it was also observed to be there in animals. So they won the Nobel Prize in 2006, Craig Mello and Andrew Fire, for their pioneer work with the C. elegans system. Okay, and again, they observed this RNA interference actually, you can say serendipitously, because they were trying to use antisense RNA. Okay, and what they observed, that basically even the control sense RNA produced suppression. Okay, so it was not something which was expected, because sense RNA should have given like higher expression level, but it caused suppression. And then when they looked into the cause, they saw that their control sense RNA was contaminated with the double-stranded RNA. Okay, so there were some antisense as a result of which the sense and the antisense kind of hybridize together to give double-stranded RNA. So later on, they kind of carried out several set of experiments to figure out that it is the double-stranded RNA which was the main suppressing agent. Okay, so this was also named as a triggered double-stranded RNA because it was triggering the suppression process, or you can say the RNA interference process. Now, this is the experiment, which has been taken from the molecular biology book by Weaver. So this is an experiment where they had in the C. elegans ovaries, okay. This was the particular mRNA, MX3 mRNA, okay, and they were using an in situ hybridization technique to detect the presence of this mRNA in the developing in this ovary. So these are like 1, 2, 3, 4 cells, okay, and control cells. And here, when you do the in situ hybridization with a probe for the MEX3, so you can see that this coloration showing the presence of these mRNA in the chamber in these cells, okay. And now in the C, they had only provided the antisense RNA. So it is kind of antisense technology, what has been in use since 1980s. So what they expected that would antisense will hybridize with sense and thereby lead to lowering of expression, and that was quite evident. So compared to this is definitely much lower. But the surprising fact was in the D. Okay, so in the D panel, what was there is the double-stranded RNA. So double-stranded RNA, that means both sense and antisense were kind of hybridized, and that was the construct which was injected into the ovaries. And here, if you see, basically that the level of suppression that you achieved with the double-stranded RNA was far, far better than what you could achieve with just the antisense RNA. Okay, and not only that, it is like spreading in different cells. So when you are injecting, probably you are injecting from one side, so it is probably here, but somehow the suppression is spreading from cell to cell, and all the cells are showing massive amount of downregulation of this MEX3 transcript. Okay, so this is the first observation in the animal cells for an RNA interference triggered by the double-stranded RNA targeting the MEX3. Okay. So now it is not exclusively in C. elegans, but it is there everywhere. Okay. So of course, in Neurospora, in Schizosaccharomyces bombay, you see that, but not in the Buddingase. So Saccharomyces cerevisiae doesn't show this RNAi mechanism, but this is not something which is unique. Of course, it is not there in the prokaryotes, but similar kind of thing exists, but not exactly the RNA interference because those components are not there. So, but mostly the mechanism has been well studied in the C. elegans system and also from the Drosophila system. So most of the machinery that is involved in this process have been first identified in these systems. Okay. So when we talk about the mechanism of the RNAi, we basically talk about the role of a very important molecule, that is dicer. Dicer, why it is called dicer? Because it seems to dice up or fragment the double-stranded RNA into small fragments, 21 to 23 nucleotide long. Okay, so because it chops off the double-stranded RNA, that's how it gained the name of a DUN. And this is a you can say a nuclease or an RNase, which is basically involved in the fragmentation. Okay. So it was first purified from the Drosophila embryo, and when it was purified, it was found to be associated with small RNA fragments, both sense and antisense. Okay. Now, what was observed that the presence of these associated RNA is very important for the dicer activity. Okay, because if you use a micrococcal nuclease to degrade this RNA, then basically you abolish the effect of the dicer, okay, to digest the mRNA. Now, what was again from different set of experiment, it was found that dicer, the triggered double-stranded RNA that is added to the system gets chopped off into fragments about 25 nucleotide long, and then these fragments can associate with the dicer, and I actually kind of provide the guide sequences that allow the nuclease to target the corresponding mRNA. Okay, so which RNA should be, so that is the guide sequence which remains associated with the dicer. Now, dicer gene is present in all organisms that exhibit RNA interference, and if you mutate this particular gene, okay, then what was observed that the RNAi effect, it gets inhibited. So that clearly suggested an important role of the dicer in the mechanism of RNA interference. So from these set of experiments, it was concluded that it is the endonuclease that degrades, okay, or chops off. You cannot say degrades, but actually it just fragments the double-stranded RNA into very small sizes of 21 to 24. And in higher eukaryotes, dicer has been shown to have the ability of kind of unwinding the two strands separately, like intrinsic helicase activity. It can separate, it can break the hydrogen bond and pull the two strands apart. So it is a member of RNase 3 family, okay, and it leaves, like after the cleavage of the big trigger RNA into small fragments, the way it cleaves, it leads to nucleotide 3' overhang and also phosphorylated 5' ends. Okay, so that is how it it cleaves the double-stranded RNA, big double-stranded RNA, into smaller fragments. Now, this siRNA was the kind of experiments that was done. So it was in an in vitro system, and they took two luciferase RNA construct. Okay, so and what they did, they either labeled the 32p labeling. So either they labeled the antisense strand alone, or they labeled the sense strand alone, okay, or they labeled both the strands, okay. And in the experiment, so they have taken the this is a kind of, you can say, the for first so here they have taken the lysate, okay, and which is showing the plus wherever the lysate has been added. The first lane is the control, that neither the mRNA has been nor the lysate, okay. So in first case, there was this double-stranded RNA, and of course, you have the corresponding mRNA. So you are looking at the fragmentation of the RNA, and you're looking at the fragmentation of the RNA and the appearance of this short truncated products of 21 to 23 nucleotide long. And what you see is, when there is like no mRNA was added, okay, still you see that the fragmentation of the double-stranded RNA, which was labeled in both the strands, you could see, because you could monitor it by radioactivity in the gel, okay. And when you compare to lane 3, where mRNA was added, again there you see the fragmentation. So whether, irrespective of whether mRNA was present or not, you see that the lysate contained this containing the dicer was able to fragment the double-stranded RNA into small fragments, that is 21 nucleotide long fragments, okay. Now here it is the same experiment, but here the intensity is slightly less because only one of the strand was labeled. So you are not monitoring the fragmentation of the other strand. So you are only monitoring, so that's why the intensity, but yes, irrespective of presence or absence of the corresponding mRNA. So it is not that mRNA is needed to trigger the cleavage of the double-stranded. So it is irrespective of the presence of mRNA. The double-stranded RNA is a substrate for cleavage by the dicer, and then you are getting these small fragments, okay, generated. Uh, so this is with just the antisense labeled thing, okay, minus plus mRNA, or this is the sense minus plus mRNA. Now, the same thing with the other Ranella luciferase construct, also the similar thing, irrespective of whether the RNA was there or not. Okay, so here RNA was added, here no RNA, mRNA was there, but the double-stranded construct got cleaved and you got these small products. In the lane 11 and 12, what was done was basically an antisense Ranella luciferase construct. So this was not a double-stranded RNA, was not infected, was not provided, but instead only a single-stranded, that is the antisense construct was provided. And if you look into here, in presence of the mRNA, of course, it was not as robust as like this, but in presence of mRNA, you see a faint band in this image, which tells you that this particular antisense goes and hybridizes with the sense mRNA that you have provided. Okay, and the mRNA that's always sense. So when and because of this antisense sense forming a double-stranded RNA, of course, it is in much lower intensity compared to this, but then that also is a substrate for cleavage by the dicer that is present in the so RNAi competent lysate. So now we know that it is having the endonuclease that can cleave, and you are getting this short. The other thing which is important is the presence of ATP. So when they depleted ATP from the lysate by using a glucose and hexokinase system, they saw that this kind of truncation was prevented. So what was shown that RNAi requires the presence of ATP for the. Next, they wanted to look into basically from this RNAi competent extract or the Drosophila extract, what they designed 3 different double-stranded RNA construct, and all these RNA construct were differing by the position by around 100 base pair. Okay, so the double-stranded RNA that is labeled C was closest to the 5' end of the Ranella luciferase mRNA. Okay, and the double-stranded RNA B was like around downstream, 100 base pair downstream to it, and another 100 base pair downstream was the double-stranded RNA construct. And they were looking at the basically the cleavage of the mRNA, okay, and they were looking at the cleavage product that they get, the size of the cleavage product that they get with these 3 different double-stranded RNA construct, and all these were labeled at the 5' end, and after the cleavage they were run on polyacrylamide gel and they were analyzed by autoradiograph because they had carried the label. So what was observed, like clearly, though the bands are not very clear to you probably in this image, but what was evident is the double-stranded C RNA, that is the construct, that is C construct, was able to give you much shorter truncated products. Okay, and this was which was kind of closest to the 5' end of the RNA. So it seems like the position where this is the double-stranded RNA, and because the trigger or the guide RNA that will be generated from this particular construct will be directing the entire cleavage machinery to the mRNA. So because it is closest to the 5' end, and that is what we are monitoring. We are monitoring the products from the 5' end because that is the one that is labeled. So we are seeing the shortest products coming from the RNA, which is closer to the 5' end. The binding site is like closer to the 5' end. And if you see that, again, the shortest product that you got with construct B is again 100, almost like 100 nucleotide difference between what you got with C. So there seems to be matching with the position that they kind of hybridize with that of the mRNA. Okay. Now, again, when they did careful analysis of on high resolution, when they resolved these cleavage products, and they tried to look into the gaps or the distance between the consecutive cleavage products, they figured out that they are cleaving. The cleavage is happening in the interval of 21 to 22 nucleotides. Okay, so most of the like bands were separated from each other by 21 to 22 nucleotide, except for a unusual band which is shown here, which is different from the other band by around 9 nucleotides. And what was seen here, that this is this unusual cleavage site lies in a range in an area of the RNA which there was 7 uracil residue consecutive, and it looked like that because of this 7 uracil residue, this area was kind of more susceptible to cleavage. But rest of the sites were separated from each other by this interval of 21 to 23 nucleotide. Okay. And not only that, when they kind of sequence these cleavage products, in out of the 16 cleavage site that they could map, 14 were at the uracil. So there seems to be a preference of cleaving the mRNA at the end of the uracil. Okay, so this clearly said that the truncation happens at the end, or the cleavage happens in the interval of 21 to 23 nucleotide. Now, this is the simple model for the RNAi that has been put forwarded. Okay, so you start off with the double-stranded RNA that you introduce into the system, and then there is dicer present inside the cell, which binds to this double-stranded RNA, cleaves, and then cleaves the double-stranded RNA into this small fragments. That is the 21 to 23 nucleotide, and then there is either ATP dependent helicase or dicer is capable of doing this process. The strands are separated and you have dicer associated with these complexes. Okay, and then this complexes now form what what we call as active siRNA complexes, or you can say RNA induced silencing complexes. Now, for the cleaving the target mRNA, okay, what was observed that it is not dicer which does that, but there is another protein, at least in the Drosophila, what they kind of identified is this protein Argonaute, which is called slicer. Okay, so that is part of this RISC complex, RISC complex, which is important, plays an important role in kind of taking this to the mRNA and then triggering the cleavage of the mRNA. Okay. Now, basically, dicer from higher eukaryotes was shown to have this helicase activity, but not the initial protein that was crystallized from Giardia that did not have the helicase. But this RISC complex was shown to contain another nuclease, okay, that is not dicer, that is the Argonaute. And that is the active siRNA complex. And this is the small interfering RNA that you are talking about. So that is the siRNA, and which is associated with this protein, which will be capable of going to the and binding to the target site, driven by this hybridization between this RNA and the sense RNA, and then there will be cleavage. Okay. Now, the Argonaute-like protein, so initially no eukaryotic ago or Argonaute was kind of crystallized, but for an archaea, Pyrococcus furosus, this had an Argonaute-like protein, and when they did the crystallography, they looked for the presence of the domains. They saw that it has several domains, out of which this PV domain, PV domain is like it is something which we see pi RNA, which is, again, we will not discuss it, but it is involved in the transposon silencing in Drosophila. So that had this domain which resembled an RNA's H domain. Okay, now RNase H is an enzyme which is capable of binding to DNA RNA hybrid and specifically cleaving the RNA. Okay, so similarly, this PV had that domain, okay, which is capable of which showed similarity, structural similarity with the RNase H domain, and especially the presence of the 3 acidic residue, 2 aspartate and glutamate. And if you mutate this residue, you basically can prevent the cleavage that is done by the ago like protein. Okay, it had a middle domain, a PV domain, and a PAS domain. Okay, which are again the domains are found in agoprotein and the other proteins which are seems to be the part of the RNA silencing machinery. Okay, now that binds to these, so you have this RNA and you have this ago binding to that guide RNA. Okay, and then that is brought, and because of the complementarity, they kind of hybridize. Okay, and this after the hybridization, okay, so it is like a crescent shape, okay, crescent moon shaped like surface to which the RNA is bound. Okay, and then this is kind of the 3' end is kind of captured by the PAS domain, and it is such the way it has been positioned that in such a way that the target is almost cleaved in the midway, like, okay, based on the binding. So this is around 21 to 23 nucleotide, and the way it is positioned, that almost like opposite to the 10 or 12 nucleotide of the guide RNA. So the strand, that the position that is opposite to that, almost midway, it gets cleaved. Okay, so that is what was seen, and of course then they kind of looked for this Argonaute protein which was there in the Drosophila. They now wanted to see what about in the higher eukaryote, and they kind of higher eukaryotes have 4 ago 1, 2, 3, 4, out of which the ago 2 was found to have the similar kind of slicer activity, and that ago2 was expressed in bacteria. So that is a recombinant human ago2, and then they were so this was an in vitro assay where they kind of mixed with the 2 siRNAs. Positions are shown. So this is the mRNA, S500 is the mRNA, and they were 2 siRNA targeting towards targeting to 2 different regions. Okay, one towards the 3' end, one towards the 5' end. Okay, so the target is 500 nucleotide RNA, and of course what was shown is that magnesium is important for the cleavage process, because if no magnesium was added in the reaction mixture, no cleavage was observed. But in presence of magnesium, both with siRNA2 and as well as siRNA1, you saw the cleavage. Okay, and if you look into the sizes were different because the positions of these 2 siRNA were different. Okay, and they kind of the labeled thing could be isolated, and the sizes, based on the sizes, based on these markers, you can actually predict the position at where the cleavage could have happened. Now the question was, okay, so we know that the first dicer gets to the double-stranded DNA, double-stranded RNA that is introduced to the system, then it cleaves, then this forms whatever is called the RISC complex, which is then targeting to basically the mRNA for cleavage of mRNA. But how this process was happening? So again, most of the studies were done in the Drosophila, and what was based on different in vitro studies, what was shown that basically the dicer, so dicer is here DCR2, that most of the names are from the Drosophila proteins, Okay, similar homologs are present in the other system. Now, this has other protein associated with it, which has been named R2D2. In fact, dicer, though it is capable of dicing up or breaking the double-stranded RNA into small fragments, but after fragmentation, it is not capable of associating with those fragmented RNA in a very strong way, and it needs assistance from R2D2. So the R2D2 has a double-stranded RNA binding domain, and because in complex with dicer, this protein, okay, binds to this cleaved double-stranded RNA. So the dicer basically cleaves, as I said, it leaves 3' overhang, 2 nucleotide overhang, and a 5' phosphorylated ends, but then it remains bound to these with the help of this R2D2. Okay, and this plays a very important role in kind of carrying the sliced um, RNA particle from after the dicer has cleaved to the stage where AGO2 is going to bind. Okay, so this is basically an important player in what is called as the loading complex. Okay, now the function of the loading complex is basically to bring it, and also in Drosophila there is another protein called aromatase, which has also been shown to be important and could play a part important role in this loading complex in transferring the thing from this to the RISC complex. Okay. Now, once the AGO2, so it kind of, you can say that it is carrying this and transferring it to the AGO2. Okay. Now, the AGO2 is basically what it does. It kind of uses its slicer activity. Okay. So it is also having that because of the RNA's H domain, it has the slicer activity, and using that slicer activity, it cleaves off the passenger strand, okay, in this top strand, okay, into half. And after this cleavage, it also weakens the base pairing between the passenger strand and the guide strand. So the passenger strand leaves, and now you have the AGO2 remaining bound only with a single strand, that is the guide strand. And then this guide strand bound AGO2 will be directed to bind to the sense mRNA, okay, and then it will be cleaving that particular mRNA. So it is not, what is not shown here, that along with AGO2 in the mature RISC complex, there are other proteins that are involved, okay, but this is the main slicer which is involved in the cleavage of the mRNA. So now there has been from the double-stranded RNA, how do you determine which will be the passenger strand, which will be the guide strand? So again, several studies have shown, like when this kind of the double-stranded RNA is bound by these pre-RISC complex, okay, so the one side you see the dicer being attached, the other side is like the R2D2 being attached. So what has been shown, that usually the strand where the 5' end is bound by the dicer, here the 5' end is bound by the R2D2, and here the 5' end is bound by the dicer. So usually that strand which is bound by the dicer at its 5' end, that strand acts is basically becomes the guide strand. Okay. Now, another thing, that in certain organism, what was shown, that even if you start with very few molecules of double-stranded RNA, you get a very, very profound response, like how we saw in case of the oocyte. Okay, like just with few molecules you get a much elevated response and down regulation of the gene, and it spreads to all the cells and whole organism shows the RNA interference. Now how is it possible, how these few molecules can trigger such a robust response? So to address that issue, again, there were studies conducted in C. elegans, and what was observed, that one protein was observed, which is the RNA dependent RNA polymerase. Okay. Now what happens, what was again a series of studies was conducted, and what was found that once this dicer cleaves the trigger RNA and into small fragments, followed by unwinding, and you have these antisense and sense. Okay. So what happens is some of these antisense, okay, can act as a primer for synthesis. Okay, they can bind to the target mRNA, and that can be grabbed by these RNA dependent RNA polymerase that is present in the system, and that RNA dependent RNA polymerase can copy the primer, extend it further, and can generate more double stranded RNA, and this can again serve as a substrate for the dicer cleavage, thereby increasing the siRNA pools. Okay, so that way, although you start off with say one or two or few molecules of double-stranded triggered RNA, but in the system there is an inbuilt amplification method using this RNA-dependent RNA polymerase, which results in amplification of the siRNA, and that's why even with few molecules you'll get such massive downregulation spreading all over into the entire organism. And also what was observed that for this priming event, RDRP very important. So if you mutate the RDRP, you can actually block this amplification process, intensity of amplification. Not only that, if you somehow modify the antisense strand, okay, then so that doesn't it cannot prime the synthesis, if such kind of modifications are made that can which prevents it from acting as a primer, then again what you see is the robust RNA interference that is observed can be blocked. Okay, so which again tells you that because of this priming that is happening, because of the RDRP mediated amplification and creation of more double-stranded RNA in vivo, you are getting this kind of robust siRNA or RNA interference. Now, this is again shown that it is not something which is restricted to C. elegans only, but even in like plants, you have this RDR or the RNA-dependent RNA polymerase. But normally in the normal RNA, okay, because of the presence of this cap structure which is bound by cap binding proteins, because of the poly A tail that is present in the normal mRNA bound by poly A binding protein, access to RDR is not happening. Okay, and that's why you don't have any silencing. However, if there is some aberrant RNA because of problems in the transcription, if you have aberrant RNA made which lacks this poly A binding or poly A tail or poly A binding protein, then the protection is gone. Okay, and because the protection is gone, RDAR is capable of basically using this as a template. Of course, it will need so it will be able to generate a antisense RNA, which will be converting this sense RNA into a double-stranded RNA and thereby generating a substrate for dicer-mediated cleavage, which can bring into the RNA silencing. Also, if due to some viral infection or some kind of infection, you get somehow small antisense RNA being made inside the system, then again, even if you have this kind of normal transcript, protected transcript, but because of this priming event, now no longer you need this like aberrant RNA, even with a normal RNA, because of the presence of this primer, the RNA dependent RNA polymerase can extend it, and you can get a double-stranded RNA, which will be again a substrate for dicer-mediated cleavage, leading to RNA silencing. So this amplification and silencing of RNAi by the RDR was also observed in plants. Now, we move on to a different aspect of the RNAi. So whether it is always like post-transcriptional. So what was observed that this can also give rise to transcriptional gene silence. Okay, especially in the area of like the centromere, where we know centromeres are the region where you have heterochromatinization. Okay, so that is like centromeres, also the telomeres, there you have silent chromatin structure. If you place any gene in that centromere region, then what you see, those genes are get silenced because of the positional effect. Now what was observed, that when in a particular system, all the proteins that are involved in the RNAi mechanism, like RDRP, like DICER, like AGO2, they were like mutated. And what was observed, that some transgenes, when they were placed in the centromeric heterochromatin region, in these cases, in these mutants, suddenly you see that those transgenes, instead of getting repressed, they are heavily transcribed, as if there was, which looked like that because of the deficient system, RNAi's mechanism, somehow the heterochromatinization of the centromere is not happening. As a result, the transgenes that have been put in the centromeric region in those mutants were heavily expressed. When they tried to look into the transcription at the centromeric region, especially the repeat region, CEN3 repeats, what they saw that in this particular wild type cells, you hardly have any kind of transcript, but in the mutant you had lot of transcription happening from there. Okay, so what was again a series of they did chip analysis, okay, chromatin immunoprecipitation analysis, to monitor the presence of the epigenetic modifications that we have discussed are associated with silent chromatin. So we know that silent chromatin is associated with lysine 9 methylation. Okay, and lysine 4 methylation is kind of marked for an active chromatin. So in these mutants, what was observed, that those areas were associated with a lot of lysine 4 methylation, which is a mark of an active chromatin, and they were much low, okay, compared to the wild type cells. They had much lower levels of lysine 9 methylation, which is a mark of a silent chromatin. Okay, now again, what they had observed, that basically this outermost centromeric region, okay, gets transcribed. Okay, now it gets transcribed. There are a lot of reverse transcripts available. Okay, and very few like the forward transcripts. And of course, either this is generated by the RNA pol 2, or you can have RDRP, that is RNA dependent RNA polymerase, which can use the reverse transcript from that area as a template and can make the copy of that, so the forward transcript. Ultimately, what happens is, from this outermost region of the centromeric region, you have a double-stranded RNA being made based on the presence of this RDRP. So this is what happens in the normal scenario, in the wild type scenario, when you have the efficient RNAi machinery working, including the RDRP. So you will get this double-stranded RNA. Now this double-stranded RNA will be substrate for, again, the dicer, which is functional in the wild type cells. And then it will basically cleave this double-stranded RNA into short fragments, 21 to 23 nucleotide long. Then we know that the RLC loading complex with RTD2, it will be recruiting this and bringing the AGO1, okay, which will be then kind of the two strands will be separated and it will be binding to this. And there are other proteins also part of this. This is called RNA inducing, RNA induced transcriptional silencing complex. It is not the RISC complex that we are talking about, involving this AGO1 protein. And what it does, then it recruits RNA-dependent RNA polymerase to form what is called as RNA-dependent RNA polymerase complex, okay? And that, along with the transcript, okay, or directly can go to the DNA. Okay, that is which DNA? The centromeric DNA. So the centromeric DNA is the site where this RITS complex will be, or containing the RDRP will be recruited. Okay. Now once, because what has been shown that RDRP has capability out of all these, they have the RDRP was shown to have the capability of going and binding to this centromeric region. So this is what is thought, that this is RITS is recruited there. Once it gets recruited, it recruits the machinery chromatin modification, okay, histone methyl transferase, um, and histone methyl transferase will methylate the H3 at the lysine 9 position, and that will further recruit this proteins, that are the SWA6, okay, protein, which is important for the heterochromatin maintenance and establishing a silent chromatin structure. This brings in more cohescence molecules to achieve that high level of compact chromatin structure, and thereby this heterochromatinization spreads all over the centromeric region, and thereby all this area becomes silent. So it seems a little bit paradoxical that to silence this area, you need to first express it. Okay, so the transcription, so this area needs to be little bit active. Okay, so that you can get these forward and reverse transcript, so that you have this double-stranded RNA followed by the dicer mediated cleavage, then this recruitment of RITS AGO1 involved, and also what was shown that these transcript, okay, plays a role in guiding the complex to bind to the centromeric region. So there is the involvement of the transcript, and it seems to guide this to go and bind into the center, because there is base complementarity between this area and the RNA that is part of this RITS complex. Okay. Now the main thing is that is what happens in the normal cells to silence the centromere, and when you delete dicer or RDRP or the AGO complex, then basically you do not get this kind of thing happening. As a result, you do not have the centromere getting silenced, and that's why any transgene that you put in the centromere region shows the mark of an active chromatin structure, shows much lesser level of the H3, the lysine 9 methylation mark, and shows active transcription. Okay, so that is what was worked out. So this is just uh, whatever we discuss in the figure. So the outermost region of the centromere is constantly being transcribed to the reverse transcript, and the forward transcript may be produced to a certain extent. Okay, and what happens, this RDRP based production of the forward transcript, and that hybridizing with the reverse transcript, you get double-stranded RNA. Dicer cuts it to get you the small interfering RNA. AGO1 with other can bind to this single-stranded siRNA to form what is called as RNA-induced transcriptional silencing. Then you have the RDRP to form this RDRC, the RNA-dependent RNA polymerase complex, which amplifies the siRNA, producing double-stranded siRNA. Okay. Then this RITS through its siRNA associates with the OTR, or either by direct interaction with the DNA or through an interacts with the transcripts in that region. Okay, it is, and somewhere some artificially tethering the transcript to the RDRC showed that it can actually direct it to the particular area. So the transcript involvement is important. And then it attracts a histone methyl transferase to the OTR, which methylates the lysine 9. And of course, this histone H3 has been only shown, but you have to imagine that is whole part of the nucleosome, okay? Which in turn attracts more SWA6, which is a heterochromatin maintenance protein, and then that helps to spread the heterochromatinization. Now, this is not only this, though this work was carried out in SFORM-B, but Similar mechanism was also found that the RNAi can be involved in transcriptional gene silencing even in the higher eukaryotes, and their addition to this methylation of lysine or the histone methylation, the additional thing that we see in the higher eukaryotes is the DNA methylation at the CpG island. So there could be this RNA silencing, the RNAi machinery involved in the recruitment of the DNA methyl transferase, leading to the silencing of that particular at the level of transcription. So no RNA is being made, okay, because the chromatin structure is kind of getting changed. Now here, one thing is the RDRP is not there in the mammal, so there mostly the production of this double-stranded RNA will have to depend on the other RNA polymerase or some homologous region elsewhere in the genome can give rise to these kind of double-stranded RNA. Okay. Now, this is another study, which in vitro study, where they kind of designed a construct where the GFP reporter gene was placed under a human elongation factor 1 alpha promoter enhancer region, okay, which is called the EF2. So that is the control region from triggering the expression of the GFP reporter. And what they are measuring is the level of the transcript in the cells. So they have used a viral virus-mediated feline immunodeficiency virus, FIV construct, where they have put the GFP under the control of this enhancer, or and then they have transformed it into the cells where they are looking at the level of transcript. Okay, now they have generated siRNA, which is targeting the control region, that is EF52. Okay, the control region, that is the promoter enhancer region of this gene. So they have a siRNA, this particular siRNA, which is directed, designed to bind to this region, and they have another siRNA construct which is targeted towards the coding region of the GFP reporter gene. And using these two different siRNA constructs, they are studying the level of repression of the transcription. So they are looking at the mRNA expression profile, okay, the reporter assay by using reporter assay. And what they see that compared to the control, which has been normalized to 1, okay, the siRNA targeting GFP, they got around 78% reduction, whereas the one that is targeting the control region, they got even much more robust inhibition of transcription. Okay, so this was at the level of transcription. So this reporter assay, you cannot be sure whether it is at the level of transcription or whether it is at the level of translation. But then they have done some they have actually looked into the transcript. Okay, so we will come to that. So now they used, so the control region, again, we saw that in case of Espombe, it was mostly the methylation. Okay, so methylation of histone methylation that they were monitoring. So in because it is like in the higher eukaryotes, so we have this provision of both DNA methylation as well as the histone methylation. So there are two drugs, trichostatin and 5-azacitidine, which can inhibit the histone. So the TSA can inhibit histone methylation, azac can inhibit DNA methylation. So in presence of these drugs, okay, when they carried out the similar assay, what they see that the repression that they achieved with the siRNA that was targeted towards the GFP coding region, that was still effective. Okay, it was 0.22 here, it was 0.14, so they were almost comparable. However, the repression, the massive repression that they obtained in case of EF52 targeting the control region, here that was completely reversed. It was even much more like higher levels they got compared to the control. It is like 1.95. Okay, so seems like the repression that they got with EF52 was because of the control region, okay, that was getting methylated, and also heterochromatin formation was happening because of the presence of the histone methylation. Okay, so if you block those mechanism, if you block the histone methylation, if you block the DNA methylation part, then even though you are using an siRNA, you are not seeing any repression. So this again tells you that here the siRNA is working at the level of changing the chromatin structure, changing it to heterochromatin structure, and thereby repressing it. And when you block that reaction from happening, transcriptional silencing from happening by inhibiting the DNA methylation process or histone methylation process, you can revert back the transcriptional silencing that you could achieve with the siRNA. Now, this is a nuclear run-on assay where they measured the transcript level. Okay, so nuclear run-on assay is basically you first chill the cell to arrest the process of transcription, and then you break open or permeabilize the cell and you add a UTPs, and what happens is, and you incubate it for a very short period of time at 37 degrees, so to monitor. So so no new transcription happens, but what happens is already initiated and arrested transcription machinery will incorporate these radio-labeled nucleotide, and that will be basically monitoring the presence of the RNA transcript, and you can actually transfer it onto the membrane and probe it with a specific DNA probe corresponding to the gene of interest, and thereby you can actually measure the level of RNA that was there in those cells when you chilled them and you lyse them. Okay, so here what you see that using the EFF2 siRNA, you could specifically downregulate the transcription of the GFP. Okay, so no transcripts were being made. So it was not a post-transcriptional event. It was rather a transcriptional initiation process was not happening here, and the control gap DH was not affected. So this was very, very specific siRNA towards targeting the control region of the GFP construct and not binding anywhere else in the genome. Okay, so that again showed the transcriptional silencing that can be achieved with the siRNA. So again, another way to directly demonstrate that the area was methylated was using an enzyme which is methylation sensitive. So HINP1 enzyme is basically it only can cleave a DNA which is unmethylated. It cannot cleave a DNA which has been methylated at the CpG sequence. Its recognition sequence has a CpG has the CpG sequence in its coordinates and its binding site, and then you amplify that area with a flanking PCR primers. So if it is cleaved, then you will not get any PCR product, but if it is uncleaved, then you will get a PCR. So the methylated plasmid as a control, you can see, because HINP doesn't work on it, you get a PCR product. But when it is unmethylated, when you add HINP1, then it digests it, and that's why you don't get any PCR product. Okay. Now they use the two cell line where one GFP siRNA was used and EF52 siRNA was used. So in case of GFP, since the control region which had this HINP1 site, so the control region was unmethylated. Okay, so that was subjected to basically cleavage, and because of the cleavage, no PCR product would be visible. But in the siRNA transfected cells, because this area was methylated, okay, CpG island was methylated by DNA methyl transferase, that's why HINP1 could not cleave it, and because of which you got PCR product. However, if you block the DNA methylation using these drugs, then of course what you will see is no PCR product, because here the DNA will be cleaved, so the PCR will not work. So this again further demonstrated that the control region in case of this siRNA was basically getting methylated, and that's how it was getting silenced, and that's how no transcription was happening in this particular scenario when this siRNA was used, and it was at the level of transcription and not post-transcriptional event. Now, after like talking about this RNAi, so everywhere we talked about the introduction of double-stranded RNA, that is the trigger RNA, starts this entire process. But there has been always a question, like why does the cell, okay, because this the double-stranded RNA you are artificially introducing into the system, but then why the cells code for or have genes coding for dicer, AGO2, RDRP, and all the other proteins that are involved in the RNAi machinery? What is the physiological significance? And what has been observed that it is mostly probably it evolved to protect the genome from the viruses. And we know that viruses have these kind of RNA genome. So it was kind of probably evolved as a system, as a defense mechanism against the viral infection. Not only viruses, but then there are some transposons and mobile DNAs which can also jump from one place to another place very randomly, thereby creating lot of genetic genomic rearrangements, and that may be detrimental for the cell. And that's how there are mechanisms, RNAi based mechanism has evolved to keep those transposons in check, to prevent those transposons from jumping from one area to another area in a very random manner. In fact, the TC1 transposon in C. elegans germ cells was found to be naturally, like it is silenced by the RNAi. And in those cases, what has been observed that at the end of the transposon, there are terminal inverted repeats. And these terminal inverted repeats forms an RNA which has a stem loop structure. So basically a double-stranded, we know stem loop, so it has the stem structure, which is the double-stranded structure, which makes it a good substrate for dicer-mediated cleavage and generating these small RNA, which can actually go and trigger the entire process of RNA interference. And also, like as I said, the cells have evolved this mechanism RNAi to protect the genome from the viruses. Similarly, there is always a host pathogen co-evolution. So there are also some viruses which have protein that suppress these kind of silencing. Like in the plant virus, you have a protein which basically interfere and the formation, like the tomato bushy stunt virus, it binds to siRNA and it prevents the formation of this complex. And then you have the RNA binding protein from HIV again, which interferes with the silencing. Okay, so like there seems to be the virus, like the how the host has evolved mechanism to protect from virus have also evolved mechanism to kind of evade the host defense mechanism. Now this double-stranded RNA in higher eukaryotes, since there is no like RDRP as such. So the signal amplification that we saw in C. elegans, like just from a few molecules, you got a very robust inhibition or robust interference happening because of this amplification that is done by the RDRP. But in higher eukaryotes, most of these double-stranded RNA constructs are kind of transient. So how to have a lasting RNAi? Okay, so basically for that these constructs were generated which were short hairpin RNA. So again the whole idea was similar to these kind of transposon, where they will be like sense and antisense part of the gene will form a kind of hairpin and will be a substrate for the cleavage by the dicer. Okay, and this will provide make a continuous supply of the double-stranded RNA in the form of hairpins, and that will allow sufficient to keep the RNAi process going. Okay, so they had by 2004 researchers had built libraries of genes encoding short hairpin RNA, almost targeting 10,000 different human genes. So this is just shown like the two construct. You can either have Pol3 promoter driving the expression of the shRNA, or you can have Pol2 promoter like the CMV strong viral promoter targeting driving the expression of this shRNA. Main idea is to put the sense strand and the antisense strand of your target gene okay, as a part of the construct of this short hairpin with this kind of loop formation. And what will happen is they will make this kind of construct inside the cell. So sense and antisense construct will be made, which will be then subjected to the whole degrading dicer mediated cleavage, followed by agoto recruitment, followed by targeting the mRNA and thereby cleaving the mRNA and preventing the gene from getting expressed. So this is where we will stop, and in the next class we will start discussing about the other, that is the naturally encoded microRNAs. Thank you very much.